For the next 2
yeah,
weeks, I will be
biofilm
covering for Dr. Andrews. So it's going to be 2 lectures. Guys, hello. It's going to be 2 lectures, and then we're going to go over the material that you guys learned for the last 2 weeks. And on Thursday we'll have an exam, okay? And it's going to take place in 2 rooms because their arrangement not quite perfect, so we're going to split people into 2 rooms for the exam, okay? So in the previous, can you hear me? Yes. In the previous lectures, you guys were learning about vesicle trafficking, and today we start quite complicated set of material, and I'm sorry I have a lot of slides. So we are not going to cover everything today. I will split the lectures into 2 days, so we'll cover the rest of today's lecture on Thursday, okay? So cytoskeleton and molecular motors. You have listened to the talk of Ron Vale, who very nicely covered material for explaining how the motor proteins, particularly kinesin and its relative myosin, works in cells. He didn't cover in part 1 dyneins. So if you're interested, you can watch his part 2 and part 3 lectures, where he will talk more about dyneins and other aspects of the motor proteins and cytoskeleton. So what are the functions of the cytoskeleton? It generally provides cell shape and quite essential for cell motility, organization of the content of the cells. It also connects physically and biochemically the insides of the cell, the cytoplasmic contents of the cells with the external part of the cells. And also generate various forces within the cell to enable cells to move. So if we look at the different types of the protein filaments found or constituting the cytoskeleton, we will find generally 3 different major groups. Microfilaments, which is basically organized from the single subunits of actin, which is small globular protein. And the second group is microtubules. Alpha beta tubulin dimer will be the single unit that will be building block of this very important type of the filament. And the third group are the intermediate filaments. And all of these types of the filaments play quite critical, essential role in the life of the cell, any kind of type of the cell. So how the cytoskeletal proteins organize? They usually form this very stable multi-subunit polymers formed into this very stable fibers. And if you look at the individual bonds between the individual subunits, they're quite weak. They are non-covalent bonds, right? However, when they form into this polymers and fibrous structures, they are quite stable. If we lose one individual subunit from such structure, this entire fiber is still going to persist. It's not going to collapse. And this kind of loss of the individual subunit will be easily replenished through various biochemical means. So as I mentioned before, we have 3 different types, and 2 of them, microfilaments of actin and microtubules, are organized into the polar structures. What does it mean? It basically has a directionality when it forms. It's a head-to-tail formation, and it's quite important for the interaction with the various motor proteins associated with these fibers. On the other hand, intermediate filaments are not non-polar in structure. They usually organize tail to tail, head to head, form this multi-layered fibril networks. They are very symmetrical a lot of times, and they don't associate with any of the motors. Here's an example of typical organization of the cytoskeleton in polarized cells. This is a case of epithelial cells. If we look at the structure of such cell, it's very diverse and complex. You can see actin filaments on the periphery of the apical side of the cell layer. And then we see quite extensive network of the intermediate filaments which connect neighboring cells through this special organized structures on the side of the cells, and also at the bottom forming connections with the basal lamina between the cells and the bottom side of the cell. So this is through the basal side of the cell. In addition, we have a network of the microtubules, which are organized in a very special way as well. Here, for the polar cells, microtubules are kind of constitute the formation from apical to basal direction, where the minus ends of the microtubules are pointing toward the apical side of the cells, and the plus ends of the microtubules are facing the basal side of the cells. So when we look at the dynamics of the cytoskeleton, during the cell division, for instance, there's a massive reorganization of all of the cytoskeletal components. Here is an example of the reorganization for the actin filaments and microtubules. And as you can see, during the metaphase, the actin networks will form these structures at the cortical area of the cell. At the same time, they will start forming this contractile ring in between the two future daughter cells. Whereas microtubules will undergo a very specific formation of this very sophisticated network of bidirectional structure, which will hold the newly formed chromosomes in between two daughter cells, kind of pose them for the separation on both sides of the dividing cells. As you can see, as the cells progress into the telophase, the contractile ring develop into this very tightly organized ring structure. And as the cell complete the division, the network of actins and microtubules are quickly reorganized yet again to form a regional kind of steady state of the non-dividing cell, where the actin filaments will be forming various bundle or fibrous structures, dendritic structures throughout the cells, within and on this under the surface of the cells, and microtubules will be organized into this centralized array around the centrioles. So this is one of the examples of very large changes of the cytoskeleton and the cytoskeletal dynamics. Another very good example of such dramatic changes during chemotaxis. What is chemotaxis? It's reaction of the living cell to the external chemical conditions. So the cells will become attracted to nutrient molecules, or just the opposite, it will be kind of repulsed from dangerous chemicals that may present kind of life-threatening event for them. So in this case, once the cell senses rich nutrient, the actin filaments quickly disassemble its filamentous structures at one side of the cell and quickly diffuse the monomeric globular actins throughout the cell. And now these monomeric structures will be able to assemble into the filaments quickly and rapidly, on the side where the nutrient is presenting itself, and this way the cell can start moving toward the source of the nutrition. Here's some examples of neutrophils chasing a bacteria, and you can almost feel like they are intelligent beings. They are really coming after this bacteria and trying to engulf it very rapidly and right on the cue. Here's another example of amoeba chasing yeast cells, just trying to engulf this little yeast and moving after the escaping yeast and trying to go around. So you can see a lot of movement and at the same time rearrangement of the cell around to engulf this little yeast. So what fundamental features we observe and notice from this from this above slides and images and videos? First of all, we can see there is changes in distinct changes in cell shape. There's a polarity in the cells, so the cells are not perfectly round. They are not distributing their materials throughout kind of homogeneous, creating homogeneous environment. On the contrary, they are positioning number of internal elements at certain parts of their body. There's a lot of plasticity within the cell. So cells are constantly, they are not static, they're constantly reacting to the surrounding environment. There's a lot of motility. Certain cells can crawl, certain cells can move and swim. Of course, some of the cells, particularly cells that are embedded like layers of the cells, they cannot move much, but they will be reacting on the surface with the extracellular environment. If we look at the crawling cells, there's a lot of cell adhesion going on. So there's certain specific structures, protein structures that are formed on the surface of the cell and that are supported within this cytoplasmic compartment of the cell that support this adhesion very efficiently. We will talk about elements that are responsible for the adhesion also in the lecture for the extracellular matrix. We also see the dramatic changes in the shape of the moving organisms. So in case of amoeba, in the case of the small white blood cell, neutrophil, and constantly adapting to the environment. And these are features that are conserved throughout different kingdoms. You can see similar events taking place in amoeba, and similar events will take place in neutrophil. So why do we study cytoskeleton? It's basically integral part of the quite vital cellular events. Cell division, cell growth, cell survival, it absolutely depends on cytoskeleton, and mutations that often are presented through various debilitating diseases or even lethal, embryonic lethal mutations. They are quite serious and critical, and a lot of laboratories are basically looking into this type of mutations, and these are potential targets for both cancer research and also for gene therapy. So if we look at the broad responsibilities for different types of these filaments, what role they perform in the cells, microfilaments, the actin filaments, regulate generally cell shape. They are responsible for forming various cell protrusions like filopodium, lamellopodium, and this usually happens during cell crawling. There's also formation of microvilli, which are more permanent structures on some types of the cells, such as epithelial cells. Also, very important role of these filaments during the cell division, during the contractile ring formation and pinching during final stages of division. What do microtubules affect? They're quite critical in transporting organelles and vesicles, which we talked about, the vesicle trafficking in the previous lectures. Microtubules are, in fact, forming these roads for transport the vesicles, to assist vesicles to arrive to the correct membrane targets. Also, their role is quite critical during cell division in formation of this bipolar network of the microtubule array. And the role of intermediate filaments are quite critical as their specifics for establishing mechanical stability of the cell. So various compression forces that are often exerted on the cells, these filaments take care of the cell well-being and prevent cells from squashing. So they maintain cellular integrity overall, and also help to maintain the overall tissue integrity if you look at the bigger picture. So the mystery behind the motility and shape was a long-standing mystery. People tried to understand it for centuries using very simple microscopes and eventually arriving to very sophisticated single-cell techniques or single molecule techniques. A breakthrough for understanding what are the elements responsible for cell movement and cell motility was presented in 1940s by this scientist, Albert György, originally from Hungary, and he's quite brilliant. In fact, he received his Nobel Prize for the vitamin C discovery, so has nothing to do with actin or myosin filament or myosin motors. But in fact, then very soon after, he decided to switch the focus and dived into the research, trying to understand what are the molecules responsible for the muscle contraction. And in 1940s, his group discovered this actin and myosin fibril networks that in muscle cells, in skeletal muscle cells, were responsible for the contraction of the muscles. In fact, later on, he moved into the totally different subject of biophysical understanding of cancer development. So he was quite essential and a key figure in promoting cancer research. Years later, our understanding broadened quite significantly in discovering that actin and myosin networks in fact presented not only in muscle cells but in all cell types, and they just organized different types of bundles and interactions, but still cells needed to have this type of cytoskeletal components to be able to function properly. So here's an example of the cells that are either in non-stressful conditions or under osmotic pressure, and you can see that the actin networks are quite dramatically reorganized under different conditions. Here they're trying to form this kind of network lattice to help to maintain the integrity of the cells. And myosins are quite nicely localized with actins in both cases, under normal conditions and also under stressful high osmotic conditions. So let's talk a little bit more about actin. So actin as a globular unit, very small molecule, and it has ATP activity. So it is an enzyme. It's hydrolysis bound ATP, and there are generally three isoforms presented: alpha, beta, and gamma. In muscles, it's primarily alpha. And even in prokaryotes and bacteria, we can find homolog of actin, rho B, which is also able to basically form various filaments. Here's a crystal structure of uncomplexed, so single unit actin, but in ADP state. Normally, if you look at the cytoplasmic single single subunit of actin, it's associated primarily with ATP. But this is experimental conditions with ADP molecule, and ADP will mimic filamentous state of the actin. So during the hydrolysis, here's the groove of ATP binding site, and during the hydrolysis, very important loop of the actin molecule will undergo conformational change. It just flips and becomes kind of helical. This conformational change will affect protein-protein interaction. So any kind of reaction, just for you to remember, any hydrolysis will be always changing, even though you would think that it's only one little phosphate leaving from ATP to become ADP. It actually triggers quite large conformational changes often within the associated protein. And this is what drives all interactions. Here's the model of single unit that is kind of positioned or merged with the structure of the fibril observed under crystallography conditions, and it nicely correlates with the kind of superimposes with the crystal structure of the fibril. So if we look at the actins, most of the time it's two strands of polymerized chains which are unidirectional, and both ends looking toward the same direction. So both ends will be plus-ended or minus-ended. So how the polymerization of actin starts, both in vitro and in vivo? It's a rather kind of a spontaneous process, but this initial stage of so-called nucleation, where the triumvirate of three molecules form initial kind of seed to build a new quickly forming filament. This is a very rate-limiting process. So initially, this single unit actin will be kind of bumping into each other and eventually forming this spontaneous triangle of the molecules. And at this point, the formation of the filament is greatly speed up. When we're looking at the fast-growing filament, the plus end will be growing really fast. And the minus end also can grow, but at a very slow rate. And this is due to the topology of the molecules that are kind of forming choo-choo train. Basically, you have a front and the tails are not topologically preferable to add new subunits of the actin. It's the plus ends that will be more ready to accept new subunits. At the same time, when we look at the rapidly growing plus ends, we'll have a lot of actins that are associated with ATP. So here they are T's. And the rest of the filaments will contain ADP molecules. And again, as I mentioned before, the structure of these actins will be slightly changed, and they will be more prone to lose the fellow subunits, whereas ATP-containing actins will be much more favorably positioned to retain the multifilamental structure. Here's an example of image of fast-growing plastine, where we initially labeled this kind of fragment of actin with the myosins, and we can observe very nicely that the very fast-growing plastine is displayed here, and very small myosin is hanging here. So what happens normally to kind of retain the filamentous structure is, as the time progresses, filament is always dynamic, and it's undergoes so-called treadmilling. What does it mean? It's basically constant addition of new subunits of actin-containing ATPs, and constant loss at the same time of subunits of actin that are bound with ADP molecules. Here's the curve, time over percentage of subunits in filamentous state. And you can see it's an S-sigmoid form due to this lag during the initial nucleation to produce this initial seed. Once this formation happened, the process of polymerization really speeds up. So it's really rate limiting step at this point. And at some point in time, under certain biochemical conditions, so we're talking about salt conditions, there is a plateau, and it doesn't matter how many more actins, single unit actins are present and floating around, the filament will be retaining this constant kind of maintenance of rate to add particular number of actins and to lose particular number of actins. So this is called so-called steady equilibrium phase. And the concentration of the actin monomers that is reached at the steady state is called concentration critical, or CC. So at this point, the rate is addition and the loss is not changed. So if we look and compare the in vitro and in vivo conditions, so in tube, we can quite easily assemble these actin filaments. They very nicely create this very nice long filament. It's a very low value, 0.2 micromolar concentration. But in vivo, the concentration is quite high. So what does it mean? It takes much more actins to create this filament. So what does it mean? That means that actin filaments are less stable and tend to retain monomeric form. Why is that? What helps to maintain this monomeric form? Does anybody know?
In the cell, there are proteins that bind actins or things.
Yes. So we call them a number of accessory proteins. They are basically helpers. And motor proteins are also part of this large family of accessory proteins, but there are much more. So here's another example of another little video of how we view treadmilling of actin in vivo. What happens here? We photo bleached this area of the cell, and photo bleaching technique is basically utilization of high laser beam to erase the light emittance produced by labeled, GFP labeled, for instance, molecules. So originally we had this cell emitting, containing actin GFP labeled molecules. So you will see a lot of fibers that are produced here and there on the cell surface, in the cortex area, right underneath of the plasma membrane, and also a little shorter structure within the cell, in the deeper region of the cell. And when we photo bleach with the laser beam, the signal disappears. But eventually, as the time progresses, the new monomers that were outside of this square will come in and become a part of this treadmilling shorter fibers. So it's a very dynamic event. So here's an example of a large family of accessory proteins that actin utilize to polymerase. And various types of accessory proteins are responsible either for assembly or positioning of the skeletal filaments within the cell. So I want to point out that one of them is formin, for instance. It helps to initiate nucleation, these initial stages of polymerization into the filaments, and also promotes further growth of the polymer. So this protein is really responsible for growth of the long filamentous structures of the actin. Another accessory protein that I would like to mention to you is a profilin, and this specific accessory protein able to interact with the individual single subunits of actin in its ATP form and bring these actins to the formin to kind of assist formin in building long filaments of actin. Here's another example of another family that regulate the rate of the polymerization. So in addition to profilin, we have various types of accessory proteins that either slow down the rate of the actin polymerization or, for instance, breaks this growing fibers into the smaller pieces. So you can see there's a huge variety to accommodate any type of structure, to create branching, to create shorter filaments, longer filaments, to slow down, to speed up. So you name it, it's all accommodated through the accessory proteins. In addition to the accessory proteins, we have a number of signaling molecules such as phosphodienosyl phosphates. You're already familiar with those molecules. And this type of signaling molecule, in addition with sometimes certain cations, for instance, calcium ions, can assist the accessory proteins in making their job more efficient, or sometimes even initiate functioning of this very important accessory proteins, for instance, thymosin and profilin, capping proteins, CAP-Z and tropomodulin totally depend on signaling from PIP2. And gelsolin, which is the severin protein, depends both on PIP2 and presence of calcium. So as you can understand, there's a lot of signaling, external signaling that take place that will facilitate the functioning of all of these accessory proteins, and as a result, the whole network of actin will react to this external signaling and generate a new sets of various filaments. So here, I would like to talk a little bit more about role of formin. As I mentioned before, formin help to initiate nucleation of actin filaments. And if we look at the structure of the formin, it's a so-called multi-domain protein. These domains are serving basically to accommodate various functions and also to create interaction sites. So when you learn more in the future lectures about cell signaling or signaling cascades, you will get to know various types of proteins which are basically multi-domain structures that, like modules that present there to accommodate various other signaling proteins to interact and to form this network. So here's an example of such a network where we have forming protein. This is inactive folded, self-folded state, where special domain, RBD domain, can interact essentially with very special raw GTPase. So you already learned about few GTPases, SAR1, RAB GTPases. Basically, GTPases, they are all structurally and functionally very similar in the way they activate it and in the way they function. Once they activate it, they often recruit it through the activation to the surface of specific membrane, creates a plasma membrane. And after that, they can interact with the target molecule. Here it is forming through the raw binding domain. And now this forming is supposed to initiate filament building. So it presents another domain, FH2 domain, which can interact with actins. And yet there's another domain, FH1 domain, where it recruits profilin, the second accessory protein. So you can see it's a very complicated network that is basically building very fast the actin filaments. Here's the crystal structure, or rather model from the crystal structure of this kind of setting where growing actin filament is interacting with a part of the formin. This is FH2 domain. These are so-called whiskers, where the rest of the other domains are sitting. We don't see the raw GTPase domain interactions somewhere here, but what we can see is profilins, which are associated with this forming whisker, and also some of them actually recruiting actins, and basically this structure is sitting and kind of putting actins on the top of the head. Here's just a video demonstrating this. So initially, formin is floating around in collapsed non-active form, and then it becomes associated with activated raw GTPase. As a result, there's a conformational change unfolding, and now formin is able to interact through its FH2 domains with actins and initiate slow polymerization. And eventually, what happens is, once the filament is kind of initiated and starts building, the rate can be very greatly sped up by the presence of profilin. So these are profilins carrying the actin ATP molecules. And as you can see, once the actin is delivered, profilins are detached and leave. So again, it's a conformational change. Only profilins that are associated with the actins can interact with formins. Once that association is lost, formins cannot interact with the profilins. And this event can take place until there's a specific protein that comes and turns off the raw GTPase. So here it is. This is GDI, which comes and associates with raw GTPase, and as a result, formin is not interacting anymore with raw GTPase, and this process is now kind of collapsing. Actin filament is kept, and formin is now collapsing on itself, and now it's becoming again inactive until the next signal molecule will come and stimulate its interaction with the raw GTPase. All right, so as you can see, to form any kind of filament, actin filament, is quite sometimes complicated process, multi-stage process. In addition to kind of various natural types of molecules. We have molecules that, so-called toxins, that can halt the process, for instance, of polymerization. For instance, we have the whole set of molecules usually extracted from various plants or fungi, which either promote depolymerization, like in the case of cytoskeleton D, which is fungal toxin, or we have phalloidin, which is extract from very poisonous mushrooms. This type of chemical prevents depolymerization of existing filaments. So in both cases, when we promote depolymerization artificially or when we promote polymerization, this is very toxic for the cells. Cells don't want to be in a fixed state. Cell for normal functioning, it requires constant treadmilling of their high order structure. And here we can see this on this model where we have very highly organized different types of actin networks that, although they are not collapsing, but they constantly undergoing through the treadmilling. So we can see a large number of the actins in their ATP bound state coming and joining this high order structure, whereas at the same moment, number of actins bound to ADP leaving this structure. So this type of treadmilling helps to be posed to be ready to kind of react to ever-changing conditions and requirements for the cell peak. So if we look at the spatial organization of these different types of filaments, we notice various types positioned through various parts of the cell body. For instance, contractile bundles, so-called stress fibers. These are organized in antipolar networks of the fibers, and they are required really to pull cell away from something. So it's a part of the movement. You need to promote movement forward, but you also need to collect your kind of tail to move along. In addition, we have gel-like networks. This is so-called three-dimensional network that helps to organize around the cell cortex, so more closer to the cell surface underneath of the plasma membrane. Lamellipodium and phylopodium, very important for cell movement forward, particularly lamellipodium. It's like one big wave of membrane with the structures underneath, actin structures that are kind of branched, dendritic in the formation. And phylopodiums are these very long extensions, which are sort of similar to the microvilli formations, but they can be quite transient. And again, to organize and maintain these structures, we have to have yet specialized accessory proteins to specifically take care of this spatial organization of the actin filaments. And these are so-called calpain homology domain family of proteins. All of them you can see quite different in length and structure, but what makes them all kind of within the family is presence of so-called F-actin binding domain, filamentous actin. So these domains are not going to interact with the globular individual subunits of actin, but rather only be able to interact with the filamentous form of actin. And you can see they can form quite different structures, fibers or networks. This is attachments to the cell surface, so any kind of spatial organization. So here is an example of a very important formation of the contractile ring during the cytokinesis. This is one very important spatial organization of the actin filaments during the cell division. This type of organization of the actins assisted through a specific GT phases. Again, it's a cell signaling event, and only during that time this actin filaments organized to create the contractile ring to complete the cell division. Here's another example of microvilli within the intestinal epithelial cell. So here we have the actin fibers that are organized in parallel, very nice arrangement with the specific distance in between each other to create this very unified shape. And again, there's a number of different accessory proteins that maintain this actin filaments. These are quite long-term organizations, so cytoskeleton can be quite dynamic, and very transient structures can be maintained, can be forming at any given moment. But at the same time, there's a lot of structures that are persistent throughout the cell life of the cell. For instance, some of the microvilli can form and retain its structure throughout the cell life. Here is another example of spatial organization of the cortex. Cortex is an immediate lining of the plasma membrane of any given cell. And if we look at the red blood cells, first of all, red blood cells became quite useful tool to study plasma membrane and the cortex, because for the simple fact of lack of various organelles in this cell. So as the red blood cell matures, it loses its organelles. It's rather short-lived after that. And through various techniques, we can get rid of the internal contents of the red blood cells and look at the very nice membrane. So there's no complications from ER or mitochondria. There's nothing but the plasma membrane. If we look at the arrangement, the spectrin is the accessory protein that, in collaboration with the short actin filaments, form this very nicely organized cortex lining. And this network is associated with a number of transmembrane proteins to hold this network very tightly. The function of such network of the proteins is to maintain the cell shape. When the red blood cell transfers from the larger arteries to very tiny capillaries, it has to really squeeze through, and as the process goes on, it changes ever slightly the shape. So in order to maintain the shape after it passes through the tiny compartments of the microcapillaries, it requires presence of this network of the cortical cytoskeletal structure. So it acts basically like a spring. As soon as the cell passes through the smaller space, it's able to kind of push back and assume normal red blood cell shape. In addition, since it's very tightly associated to the surface of the inner surface of the plasma membrane, this type of lattice can corral number or kind of retain number of different transmembrane proteins that are sort of becoming trapped within this very localized membrane domain. So if you remember lectures about rafts, or when you will look at the cell signaling, when very specific types of assemblies, multi-protein assemblies, signaling assemblies, will be organized transiently at very specific sites. These cortical lattice really helps to retain a particular transmembrane protein in place. So although the membrane itself is very fluid and transmembrane proteins kind of floating back and forth, but they are retained within this cage. Here example of the filament-induced formation of the actin 3D network, and as you can see, this type of organization helps to establish more volume, kind of three-dimensional volume consisting of these fibers. And of course, many times we require an anchorage of actin filaments to the plasma membrane. And here's one example of the ERM protein, which will be able to, again, through the F-actin domain to interact with actins and form yet another interaction with the transmembrane protein and anchor this actin filament right at the surface of the plasma membrane. So that was actin. So how about tubulin? This is units of formation for the microtubules, and usually we have alpha beta tubulins forming this dimer, heterodimer. Both of the subunits are in fact contain GTPase activity. They both able to bind GTP, not ATP but GTP. But only beta subunit able to hydrolyze bound GTP. The alpha subunit permanently retains this subunit of ATP, this molecule of ATP. Here's the model from the electron microscopy of the alpha and beta, and the interactions between these two subunits always non-covalent. So here's a non-covalent interaction, and filaments are also formed for the non-covalent interaction. During the polymerization of the microtubules, we form this very nicely organized fiber, tubular fiber containing of 13 protofilaments. And what's so special about microtubules, just like actins that are thread-milling, microtubules also have specific events that are very specific to their dynamics. They undergo dynamic instability, and here is the
continually grow from the centrosome added to a cell extract. Quite suddenly, however, some microtubules stop growing and then shrink back rapidly, a behavior called dynamic instability.
So, as you can see, it's also this process of dynamic instability, just like thread-milling, is connected to the state of hydrolysis. So whether or not individual subunits of tubulin are connected with GTPs or GDP molecules. As the rapid growth of microtubules continue, usually at the very end, at the so-called plus end, we will find GTP-associated tubulins. But at some point, hydrolysis catches up, and there's a so-called catastrophe event, and it's a very rapid collapse of the growing microtubule. And this catastrophe happens due to a change in topology of the overall fiber. As the hydrolysis takes place, there is a curvature. The fiber becomes a little bit topologically distorted, so it's not able to maintain very nice straight line. And as the curvature grows even more and more stronger, at some point there is a collapse, and tubulins basically disassemble. And it can shrink very rapidly and very dramatically, because basically only the cap contains, within the microtubule contains the GTP bound molecules. So there's a lot of tension within the microtubule itself. So as long as there is ATP bound tubulins present at the very end, at the plus end, the microtubules are not collapsing. But once the very end undergone hydrolysis, there is a collapse. Just like in case with actin filaments, there's a number of toxins that also can affect microtubules dynamics. And a lot of this toxins actually quite useful both in research and in cancer treatment because microtubule dynamics is quite important during the cell division. So as a result, if we target cells that are rapidly dividing, and mostly it's immune system plus cancer cells, we are basically will be able to target cancerous cells very well by this type of toxins. So one example is Taxol, which extracted from Pacific yew tree, and it's actually utilized for a variety of cancers such as breast, lung, ovarian, and pancreatic cancer. So it's a very significant chemical in medical research. Colchicine is another example of chemical utilized or toxin utilized in medical treatment of different types of diseases. This is anti-inflammatory agent primarily, and it's utilized greatly in treatment of such diseases like gout and pericarditis. In research, we use synthetic drug nocadazole, which promotes depolymerization by basically binding to tubulin subunit and preventing tubulins to organize into the fibrils. So just like in case of axin, we talked about specific organizations of actins in certain structure and where these structures are found within the cells. Microtubules also organized in particular orientation and in particular way depending what's happening in the cell. So if we look at non-polarized animal cells, any typical animal cell, we will find that all the microtubules kind of organized in this star-like arrangement with the center containing centrioles. This is so-called microtubule organizing center, or mTOR. This is where the centrioles are locating, and this is where the kind of center for division will start at the same time. All of the microtubules are pointing with the minus ends toward the centrioles, and the centrioles themselves also contain, basically consist of very specialized type of microtubules. The arrangement is, instead of 13-fold, they are 9-fold triplets. So very short, little stubby structures that kind of maintain themselves as a center of gravity for this mTOR. And usually it's one center per normal garden variety animal cell. Why minus ends? Because there's specific structure that is formed during the microtubule formation, and this is where the gamma tubulin coming into play. Original kind of initiation point for the growth of microtubules will involve gamma tubulin, and then we'll have alpha and beta coming, and basically normal polymerization of tubulin microtubules will occur. And if we look at, if we try to artificially cut the cell in different parts, microtubules will require this kind of central gravity. They will self-organize. They will try to self-organize into this astral structure, even without the presence of centrioles. So to kind of maintain integrity of the microtubules really, and position plus ends toward the periphery of this kind of defective cell. How about polymerized or polarized cells? If we look at any kind of lining of epithelial cells, we will find quite different arrangement. These are non-dividing cells at this point, and these microtubules arranged slightly differently. The plus endings now pointing toward the basal lamina of the cell, or toward the basal part of the cell. whereas minus endings are pointing toward the apical part of the cells. And this arrangement helps also to promote various trafficking that happen within the cells across the cellular space from extracellular apical part to the basal part. So just like in case of axons, we'll have various numbers of various accessory proteins that will help facilitate formation of the microtubules, and also to rearrange them and form them into different types of arrangements. Here is an example of opposing action of two accessory proteins, XMAP215 and kinesin 13. So kinesin, in fact, is the kinesin motor protein. But here its function is not to traffic things on the microtubules, but rather to pry away this ends, the plus end of the microtubule. So it's a catastrophe factor. It facilitates destabilization of the microtubules. But yet another accessory protein, XMAP215, is actually bringing single subunits or actually heterodimers and bringing them to the growing plus end of the microtubules and facilitate the formation of the microtubule network. So the rate of polymerization will be increased in the presence of the XMAP215. So in addition to cell division, microtubules form a variety of additional structures that are quite vital for the cellular survival. For instance, if we look at a variety of the protozoan organisms, we'll find so-called ciliary appendages, and all of these are, unlike microvilli, will have microtubules within them. So these type of ciliary extensions will be able to move. They are like a little motors, and this infusoria is able to move around and float like a little boat by wiggling or rotating this little cilia. Here's the example of flagella, or yet another microorganism, and these are much larger and much few in number on the cell, and they are really rotating. Again, they will contain very well developed microtubular motors in the presence of dyneins that will help to rotate and propel this organism forward. Here's another example of so-called primary non-motile cilia, and this is a very interesting type of extension. So a lot of times when we look at the cell pictures, how the cells are drawn, they may contain some microvilli, but in fact most of the cells, if not all, will have a little antenna. This is like a little organelle, and this organelle, and it's only one per cell, this little antenna, or like a tail, will again contain microtubule, and it's not going to move, but what it does, it's quite critical for sensing things around. So basically, this is cellular sensory organs. We have 5 sensory organs, like smell, touch, vision, but this is what cells have to assess what's happening. And this is very rapidly growing research currently. There's a whole slew of so-called ciliopathies, the diseases that are associated with problems or defects in function of this primary cilia in various cell types within humans. As you can see, the defects are quite broad. We can find problem with hearing, brain cognition, vision, function of kidneys, skeletal structure. So it's a very, very subtle but yet profound type of problem that takes place if we have a defect within the primary cilia of particular cell type. And last thing that I wanted to mention about microtubules, what's their major function in the cell? It's of course creating these bipolar spindles to position newly formed duplicated chromosomes and during the metaphase, during the cell cycle. So this is very highly organized structure that you will learn probably more during the cell cycle lectures. All right, we have still a little bit of time to talk about monoproteins. Of course, you learned quite a bit probably previously from your previous classes, and you already viewed a video from Ron Vale, who was quite a pioneer, one of the people who was critical in studying these molecules, the monomolecules, isolating them and studying how they function. So what are they? They are basically accessory, different type of class of accessory proteins, and they are enzymes that couple the hydrolysis of ATP with the large conformational change. So a lot of times different molecules that are associated with hydrolysis of ATP or GTP, they will undergo a small conformational change. Now these molecules will undergo very big mechanical changes. So if we look at where these motors are functioning, myosins are always associated with the actin filaments, and kinesin and dyneins are associated with the microtubules only. Here's a slide that shows a large family tree of kinesins and myosins and how they are kind of distantly related to the G protein. So these are ATP binding enzymes, and these are GTP binding enzymes. But basically, they are coming from the same evolutionary ancestor. And if we look at the myosins, for instance, we will find, of course, you are familiar with the muscle-related myosins, but in fact, there are different types of myosins that perform quite a variety of functions. So here, myosin 2, this is muscle contractant, and myosin 5 is very important for the membrane or mRNA transport. So their function in the cell is quite different, and the way their processivity is functioning as well, quite different. Here's the general slide showing the kind of a set of different types of myosins that are associated in humans, for instance. There's about 40 genes coding for myosins, and you can see that in structure, they're quite all different. The only conserved part is their motor domain. This is very conserved. Here's the example of myosin 2, how it's kind of organized in a fiber of the muscles. You can see it's forming this bidirectional, bipolar fiber containing of many myosin subunits. So in order for muscle to properly function, you really have to have a lot of myosin heads to kind of create this walking contractile motion. Here's a kind of structure, crystal structure of the overall myosin, myosin 2. This is binding site to the actin, and here's we have the ATP binding site. Whereas here is the chains that will undergo motion, mechanical motion during the hydrolysis. So how do we view or study the motility involving myosins and actins? We can code the glass slide, for instance, with the myosins, and then we position GFP labeled actins on this myosin heads, and as a result, as you may remember from the video, these fibers will be moving in particular directions. So if we look at yeast, for instance, there are three general typical classes of myosins that are fundamentally different from each other. First class will be attaching to the plasma membrane and basically responsible for the overall cellular organization, intracellular organization, control of the internal structures within the cells. It's also participating in endocytosis. The second class will be responsible for contractile activity, for the muscle type of activity. And the third class will be vesicle transporting class. So this is the myosin that will be transporting various type of vesicles and cargo through the long distances. Now, kinesins are very similar in the motor domain to the myosins, although the rest of their body is quite different. As you may remember, the sequence of myosins and kinesins is different, but the overall domain structure is very similar within the motor domains. And if you look at the kinesins overall as a family, pretty much all of them will be moving toward plus end, except two, 13 and 14. 14 will be moving toward minus end, but 13 is the catastrophe factor that I've already mentioned. That is important for the microtubule stability. So here we have a kind of comparison of the crystal structure of the motor heads between kinesin and myosin. And myosin is quite larger, but the core domains are very conserved in structure, although again the sequence is different. So only when you look at the structure, we can see how similar they are. All right, and the last slide for today, as I want to mention, is how the myosin and kinesins are moving along the filaments. Myosins really kind of slide along the actin filaments, and usually normally tightly associated with the actin filaments. And when ATP associates with the myosin head, then there is a detachment from the filament, with eventually there is hydrolysis taking place and movement of the head, and again reattachment to the actin filament. Kinesins kind of walk along the microtubules, and as you can see, one head is always associated with ATP and another is with ADP. So at any given moment, one motor unit will be always attached to microtubule. So kinesins most of the time attached to the microtubules during the walk. Myosins briefly detach and then reattach. Right? So let's stop for today, and we'll continue next time.